This video will demonstrate a method to set up high-throughput crystallization trials of purified membrane proteins using lipidic bicelles. First, bicelles are prepared by mixing a lipid and a detergent. Purified membrane protein is incorporated into the bicelle mixture, and crystallization trials are performed using a nanoliter robot. Visual inspection using a standard light microscope reveals the presence of crystals. Once formed, the protein crystals can be extracted and frozen for data collection and structure determination.
The main advantage of this technique is that, owing to the unique phase behavior of bicelles, standard equipment and robotics available in the lab can be utilized for crystallization setup.
Bicelles is such a simple technique and allows you to take your target membrane protein out of the detergent micelle and place it into a lipidic medium. This gives you an entirely new set of conditions to screen. It is such a simple method that has been successfully used on many membrane proteins that there's just no reason not to try it.
The first step in bicelle-based crystallization is the preparation of a bicelle-forming lipid amphiphile mixture. This process requires considerable effort and is the most time-consuming. Note that it may take several hours to complete.
Bicelles can form in a variety of lipid amphiphile combinations and over a wide range of concentrations. For suggested starting points, see Table 1 in the accompanying written protocol. Begin by preparing 1 milliliter of 35% DMPC Chapso mixture at a 2.8 to 1 molar ratio. To do this, weigh out 0.26 grams of DMPC and 0.09 grams of Chapso in a tube.